Before going for his formal introduction, there is a very sad news from our side that one of our very respected alumni FCTL member, Dr. Shivi Vargish, she left us on 10th of January at a very early age. She was suffering from some enlightenment. I would request all the participants to for 2 minute uh, silence for her soul, so that we pray that her family get all the strength and her close and near ones have all the belief and soul for her so that she can rest in peace in heaven. I'll just tell everyone to just give a 2 minutes of silence prayer to her soul. Thank you everyone for your kind presence. Now moving on to our webinar series. So before today we have Mr. Ashutosh Tank, who is an alumni. Before going for any further introduction, I would like to request Professor Mishra to tell a few words about him in his lab and the experience, and then I'll go for formal introduction. Professor Mishra. Sir.
Yes, Jayashree, thank you very much. And I think that today this message which we have again, you should write this message from FCTL side to husband, to her husband, all right?
Yes,
Send
sir.
on
Yes, sir.
email to Saju, Dr. Saju. In
Yes,
fact,
sir.
we lost in her a very, very brilliant food and dairy scientist and thorough or perfect, gentle personality. But anyway, everybody knows Shivaji. इनफैक्ट अभी कुछ दिन पहले अबाउट एक महीना पहले शी वाज ऑल राइट शी वाज सफरिंग अर्लियर सो शी टोल्ड सर नाउ आई एम फाइन एंड नाउ आई विल गिव द सेमिनार दिस एफसीटीएल सेमिनार बिकॉज हर सेमिनार वाज ड्यू बट एनीवे जो ऑलमाइटी को पसंद था so regarding Ashutosh tank so before I ask either Rohit or this Navneet one of them to formally give something about Ashutosh. Let me just tell yes, Ashutosh, he was here for a very brief time, I think about 1 year, Ashutosh, with us.
Yes, sir. That's
Yes,
correct.
he was there in 1 year in one project with us. And from that, he moved to uh, Cork's, UC, University Cork's, Ireland, for his PhD degree. And after getting PhD from there, he is now serving there itself. And I will only say that yes, when he was here in Ireland, he was a very silent guy, calm and quiet, चुपचाप. लेकिन आपस में I could know that इनका जो 4-5 का ग्रुप था, CFTRI group, very active group at that time and very dynamic group. So they were all enjoying the life fully. So details will be told by Navneet or one more thing, Jayashree, if now people, everybody is here, so in this 2022, I would like to take that opinion of all the FCTLs and also our senior colleagues are there, that let us expand this horizon of this seminar little further. All right. Now we can
Yes, sir, definitely.
invite our other like-minded people, senior persons or professionals, etc. Those who are at least in somewhat related to our lab, or thus in one or the other capacity. Like we can request once Professor Viswanathan to give some thing to tell something about or such other persons. So we will let us try to have these things in here, right? Okay, Jara, I am in some seminar. Yeah, I will talk later. Okay, so with this, I think let's Navneet, would you tell about
Yeah,
Arthos?
no problem, sir. So, Ashutosh, so my early remembrance was while I was pursuing my BTech. So, Ashutosh was kind of was also pursuing his Bachelor of Engineering from A.D. Patel Institute of Technology, and from then on, he went on to come with CFTRI. But as a person, he was he had that scientific temperament kind of, and he was not, I believe, satisfied with the job or whatever he wanted to do, and that's where I think Ashutosh landed up in your lab. And your lab is something like, it's like a, it opens the door to the world. It's like kind of a, I always say ki the lab of Mishra sir is something in which anybody enters, they have all the avenues, paths to go to anywhere. So that's how this lab has been. And somehow I think I would still say ki Ashutosh was lucky enough. So during initial days, he got little bit kind of confused ki where he landed up, kind of, and because there was so many things to do in his lab and everything. But as in when things unfolded, somehow I think he wanted to pursue something different, more higher studies and other stuffs. He went off, but that lab always remained close to his heart. So somehow, whatever time he spent at IIT Kharagpur IST, I think that was most cherished moment for Ashutosh. That's what we discussed while he was living at IIT Kharagpur. And then I think he landed up in Ireland, continued to do multiple places, but this cord of connection with the lab always been there, and that is what we could witness right away. So I think I would say that Ashutosh is fantastic as a human being. Uh, he had a mindset of an scientific and a scientific mindset, and that will propel him to do multiple things at its scientific level or at professional level. So, but we could not meet face to face. Unfortunately, he's been outside kind of. But whenever he's in town back in India, we should have a get together at IIT Kharagpur. That is all we can say. And additional things, I believe, I think Rohit can put up any more coincidences, because I think he shared a room. In fact, he shared room with me also while we were at IIT Kharagpur. So we had a lot of fun, Mishra sir. I can't speak about those fun at IIT Kharagpur, at least at this forum. but yes, the whatever time we enjoyed, and it's, I think we all are lucky enough to at least having gone through FTCL and then gone to multiple places in our life. So this is the common thing which connects to everyone, to us, and this makes this forum kind of a more than a family to us, kind of. Whenever we can connect, unfortunately, we could not connect due to some of our issues in Bombay and other stuff. But somehow it is a wonderful thing. Uh, it's a wonderful thing to reconnect with Ashutosh once again. He was there at my sister's marriage also, kind of. He came to my place. So it's long history, kind of, along with that. So I have both personal and professional, everything. And from my personal side, I always wish him best of luck in his life. And I'm sure he will keep the standards of FTCL to the highest level. And we all strive to do that in our life. And because that has been a blessing for all of us. And Rohit bhai, if you want to add anything more, please come up.
No, you said everything very nicely and sum it up. What I can say about Ashutosh is, as a person, I saw him as very exploratory and highly skillful person who was introvert in the beginning, but I think slowly he picked up and started, making a lot of friends. And I think he landed up in IIT, which was very good thing. And we all say that IIT is a land of dreams, and that actually opened up the opportunity for him as well. And as I said, that he was very exploratory, so that gave him a lot of confidence to explore multiple domains. And really glad that he reached to a point where he feel that he is doing good and able to contribute back. And connecting with FCTL and all the alumni is always cherishable for all of us, and so is with the Ashutosh. Ashutosh is very fond of cooking, I remember, and he was very famous among the BTech guys because he used to invite them for the biryanis and all. I still remember that we used to eat a lot of biryani together, and he was the cook. And yeah, I mean, he do a lot of things by heart, and very emotional as well, and very scientific, in his thoughts. So yeah, I mean, I wish you all the best in your all the endeavors, and whatever you do to the society and to the FCTL will always be cherishable and will be appreciated. So thank you so much, and I open this platform for you to speak today.
Thank you, Rohit and Navneet. Honestly, I'm very flattered, and I'm about to cry, but I'll hold my tears back.
You can do
Uh,
for 5 minutes and then start.
should turn off my camera first. uh,
Yes, we should start now.
yep.
Yes, sir. We should definitely start now
Yes, sir.
because I think we are all about it. So, going on the formal introduction of now, from moving on the lighter note, I think we'll go to the serious act. So, I'm introducing. Mr. Ashutosh Tank is a technical operation engineer at Janssen Sciences Ireland, which is a pharmaceutical company of Johnson and Johnson. He was part of our lab as he was a project assistant in FCTL IIT KGP. And after that, he completed his master's in food sciences and technology, as well as he has a PG certificate in biopharma processing. His career basically started as a food engineer and a food technologist who has a comprehensive knowledge. Now, currently, Mr. Tank is working as a technical operation manager, where his role involves validation activities, studies that supports the sites to comply with GXP regulation defined by various regulatory authorities like HPR and FDA. FCTL and the all audience welcomes you, sir. You can start your presentation. All the questions will be taken at the end of the presentation. By the meantime, please, apart from the speaker, please mute your audios and the questions can be written in the chat box. Welcome, sir. Sir, just you can present.
Thank you, Marisa, and thanks very much. It's actually an honor to be speaking in front of everyone, especially Mishra sir, after a very long time. So naturally, my heartbeats are going up, and with the formal introduction of by Navneet and Rohit, I think the bar has gone really up. So I'll try to maintain that. I'll start off by just saying that I want to welcome you all, and I want to thank you to take this time. And from my end, I really hope that everyone is doing really, really well in terms of their health, because as you know, every single person is affected by COVID. None is spared, and I hope that everyone is kind of recovering, and everyone doesn't face any severity of this. And it's very unfortunate that we lost Dr. Shibi, and I think I put my deepest condolences to her family and to the FCTL team. So moving ahead, I like to say something more about myself, and very briefly. And the reason I picked this is because I think many of us would maybe find some similarity of their life and their career pathways is because I come from a very small town, which is which is close to a commercial port, and I just broke that conservativeness of the parents' ambition that mera beta doctor banega, mera beta ye banega, and I said, "Nahi, I want to pursue this." Moving ahead, I became an engineer. When you move ahead and when you pursue your dreams, you feel like being a rock star, and you start having your dream jobs and everything. But at one point, you start feeling that where your life is going, and do you want to change? I think there's a speaker on. If I just request someone's mic or speaker to be just turned off, please. Can I please request to mute your mic? Oh, yeah, that's perfect. So I was saying that as at some point you feel like you want to explore more, and that's what I did by going through different career options in my life. And I was fortunate that I came to FCTL lab, followed my alumni, peers, and got their fantastic support. But at the same time, even when it gave me a huge gateway to go and explore abroad, reaching to Ireland and Cork, there were in my life, and I suppose you can compare that in everyone's life, you get hit rock bottoms every single time, maybe personal, professional, academic, and as human, we just dust off, just get up and focus on ourselves and just move ahead to reach that some bit of normality or a routine life, and that's what I am still pursuing right now. So this is just something more about me apart from what everyone said just before a few minutes. And so I just want to give a huge big greeting from Cork. That's where I am, and that's where my new home, and that's when I'm trying to find my peace. And I was saying to Mishra sir that this is the real beauty of Cork, and that's how it looks like. And if anyone is fond of Heineken, that's the small brewery that's just in the heart of the city. And I used to stay close by here, where you can every morning you can smell the fresh malt being processed and fermented. So I just want to give a big greeting. And before I proceed ahead, a really huge thank you to Dr. N Mishra. I think in my heart, I'm always in debt to him. And not only him, but all the FCTL members that I went through, maybe few BTech students who passed by. I think the small supports, motivation, encouragement, spending time together, they all contributed to where I am right now and what person I am. So I just want to say a big thank you to everyone. So I said a little bit about my personal life. Professionally, I work in Janssen Sciences Ireland. So I decided to transition my career from food and research to a little bit more professional. So I found out that Ireland is a huge hub of pharmaceutical, biopharmaceutical, and even medtech device companies. So and these companies were quite eager to accept people who have diverse skills, and the best thing is that it gives you an opportunity to train as well any candidate that comes from a diverse field. So what I did was I prepped myself, and when there was a huge rock bottom, I said, like, it's time that I feel I should pursue something that needs to be a little bit more professional, something that can cater a more professional job at this place. So that's why I moved to biopharmaceutical and pharmaceutical. It's a very promising area because as like food, which never stops, and people don't stop eating. Biopharmaceutical and pharmaceutical is something that people will not stop taking medicine for their betterment. So that is one thing that it's really promising. So my role in this company for the last 4 years is being a technical operations engineer. It's a small but a very strong department that caters to routine manufacturing. And what it does is its role, its function is essentially to support validation of equipment's process, any issues or investigations. So my role as a technical operations engineer in the past 4 years was to support, process verification reports, then cleaning validation of equipments on site, and even process validation support. So we do different studies that enable and give confidence to routine operations and the regulatory authorities, saying that it's the process is well able to keep up the standards even after repeated cycles of manufacturing. So that's my job profile. And that's which is also my current job profile since past 4 years. Just about this facility that you see, it's actually a real-time aerial view taken back in 2021. When I joined this site as such, there was a huge expansion going on, which was considered one of the biggest investments of Johnson & Johnson. And this building, I hope you can see my pointer on the screen. Can someone confirm me, please?
Yes, we can see Ashutosh.
Okay, perfect. So if you see this building here at the very end, this whole area was absolutely bare land. This was the new expansion because the company wanted to invest and double the production capacity. Including this, there were new facilities added. So there was huge work involved, and I was part of the team that was taking care of the validation of this new facility. So as such, it was a great opportunity for me to learn and also sustain myself in this industry too. So from it's a very busy site, and there are a lot of challenges coming up. And what I thought in today's presentation is that I thought that biopharmaceutical, although it's a very known term or terminology, but maybe if I can give you a very, very brief overview about what is the manufacturing, what's involved in the manufacturing, then I think that they'll kind of make justice to what I'm speaking and what I'm doing right now. So my intention for the next, say, 20 to 30 minutes would be to give you a very high overview of manufacturing. I won't go in detail about DNA or protein structures because it's not my background. I don't have biotechnology background, and I don't want to challenge the FCTL members because I know that their knowledge and their experience behind this subject would be absolutely fantastic. So I will try to make it as easy, slightly non-technical, and help you understand what's involved in the manufacturing. So maybe you can appreciate the efforts that goes in, and at the same time the challenges that we are facing, and even what I am facing right now working in the site. So moving ahead, that's what's today's menu is. It's chemical free because it's all biologics, it's all bioprocess. What we'll do is we'll look at a brief check on biopharmaceutical, what it means, the mode of action, how it's manufactured, upstream, harvest, downstream, filling. These are four main phases in the bio processing or biopharmaceutical manufacturing, and we'll see what are the current challenges that as a site we are facing, and not just our site, but as a whole biopharmaceutical industry, what they're facing, what challenges they're facing right now. So in a very short and a brief way, I'll not go much into details, but I'll just pinpoint certain areas. I'll be more interactive. I'll be speaking more, and at any point, obviously, if you need me to elaborate, just keep a note, and please do tell me at the end of the presentation, and I'll be happy to answer you or provide you with more information if I cannot actually say anything right now. All right, so let's move ahead. So what exactly is biopharmaceutical? So biopharmaceutical is just a therapeutic product created through genetic manipulation of living things. And when we say living things, it's essentially cells, essentially the core component of cells, which is DNA, RNA, and that kind of transcribes to complex proteins, which are also called antibodies. So I really hope everyone has heard the term antibody. Antibodies essentially are proteins. They are complex proteins present in your body. They are triggered by your B cells or your immune system. So if you're infected with something, the immune system is triggered, and it releases antibodies, and those antibodies try to target that cell, or say like a virus, that a normal flu that you get. So it'll try to see which antibody can actually affect or destroy that. So that's what it is. But what we do here is they try to actually engineer that antibody and say that which is the best antibody that can fight this particular disease or this particular disease cell, which has a specific antigen. Okay, so they that's how they came up with different recombinant proteins and monoclonal antibodies, and almost 80% of the biopharmaceutical company, the commercial products or the commercial biopharmaceutical drug products are actually monoclonal antibodies. Monoclonal antibodies, why mono? Because it has one active site. If you look at an antibody structure, it essentially looks like a Y shape. That's as you see on the screen. If a cancer cell, or say if a disease cell has an antigen that is has a specific binding site of an antibody, what it does is it looks for that antigen and it kind of binds correctly, and then the mode of action starts to kill the cell, or whatever the mode of action is. So the reason why it's called monoclonal antibody is because it has a specific binding site and it goes directly to the target cell. If it is something else, like say, it won't fit. So that's the whole idea behind monoclonal antibodies. And I think somewhere in the world, in some guarded facility, and different companies, maybe our company or a different research company, would be doing a lot of research to come up with strains that can actually generate these kind of antibodies. And we get essentially what we get on site is the best strain that we can actually grow those kind of cells and extract the protein. So that's done somewhere in some part of the world, which I absolutely don't know. So what I'll do is, now in the next slide, I will try to actually show you a video which will give you more visual about how the mode of action is for these kind of monoclonal antibodies. Okay, so I hope that works. So I hope you kind of got a good understanding of how the antibodies, the mechanism of antibodies, because it's really important to identify that they're very specific, and as a result of being very specific, they're actually they're targeted for more severe diseases, like different types of cancer cells or different type of cancers than arthritis. They are also targeted for certain rare diseases like Castleman's disease and Crohn's disease. I think if I am correct in saying this, some of the early bio pharmaceutical drug was actually insulin, and that was produced way back in I don't exactly know the year, but it should be somewhere in the 70s or 80s. That's when this was still prevalent, but it was really expensive, and it was very rare, or a special case to be manufactured. But I think slowly the popularity grew, and companies identified that biopharmaceuticals would be it's a booming industry, or not only in terms of as a commercial business, but also in terms of treating patients with diseases, because conventional methods of treating cancer, maybe by removing tumor cells, that's fine, but then using chemotherapy, which has a lot of side effects. So biotherapeutic drugs or protein drugs can actually help to kind of not eliminate the side effects, but I would say that with least side effects and very specific target, it could help prevent diseases. I from a company point of view, and even speaking with actual patients who had cancer and who used drugs from our company, they recovered well, and a lot of patient videos that were shown, it showcases that there's a huge potential, and it gives a hope back to patients, and in other words, with least amount of pain that people usually go through if they're doing chemotherapies. So coming back to the presentation, I just wanted to highlight the difference between our usual chemical drug and the biological drug, because I've been speaking about biotherapeutic or biodrugs, but I'll go through a brief differences between both of them, so that you know, because in routine life we take paracetamols or say aspirin, Disprin, which has a certain chemical compound. So these compounds are small. They're quite small molecules, and that's why majority of the pharmaceutical industry, they term the chemical drugs as small molecules, while protein drugs, they're quite large in size as a molecule. That's why they're quoted as large molecules. Chemical drugs are chemically synthesized, as the name suggests. So they are more stable as a drug. They're also more susceptible to heat and other adverse environments. But at the same time, if protein drugs are quite heat sensitive and they're quite unstable, so they need to be placed, or say need to be. They cannot withstand severe environments. The drug delivery for chemical compounds is usually oral in form of tablets, but these can be changed based on the application, like nasal sprays or syrups, or even lotions or creams, while protein drugs are injected directly to your bloodstream. So that comes again to a limitation of a chemical drug, and why there are more side effects is the fact that when you take an oral drug, it goes through your digestive tract, and by the time it reaches your bloodstream, it goes through all the harsh environments of your stomach, being acidic. And then there's the compounds are also lost as well. And by the time they reach the intestinal lining, when they get a gateway to your bloodstream, I think not maybe 10, 15, or even 50% of the drug is actually lost. So that's one of the reason why, when you take a small dose or small strength of medicine, you have to keep taking it after every 6 hours or 12 hours, depending on the drug. But what biological drug does, it takes an expressway. It's because it's injected directly in your bloodstream. It can actually go directly to the specific site of where the antigen is, and in that way, it avoids a lot of side effects as well. Chemical drugs are easy to manufacture because they are chemically synthesized. There's not much complex process involved. They are low cost to patients as a result. But on the contrary, biological drugs are really complex to manufacture, and because of the complexity and the sensitive nature of the proteins, the cost of manufacturing is really, really, really hard. It's really high, and as a result, it impacts the cost to the patients as well. So that's why we see that a lot of biological drugs are kind of not even seen much in various markets, except for European countries, except for US. And last is that chemical drugs, when a compound is synthesized, there's one active ingredient that kind of fights against your disease. That active ingredient has a certain a patent life. So after, say, 10 years or 20 years of patent life, it kind of becomes available to all the pharmaceutical companies, and because of that, a lot of generic medicines are available with the same active compound. So that's why you see a lot of companies, you'll find the same different type of drugs. If a doctor has prescribed, say, one medicine, you go to the pharmacy and say, like, oh, we don't have this, but we do have this. It's exactly the same. The reason behind that is because there is a generic available that the companies can manufacture. For biologics, it's the same. They are called biosimilars, but it's really difficult to define the complex structure of the protein drug. But there is a time when probably in near future, within, say, 10 years or 15 years, that biosimilars would be in the market, and that will kind of help to even reduce the cost to the patients. So that's the main differences, or I say the key differences between chemical and biological drug compound. Another reason that I said why biological drugs are quite expensive is because of the fact that they are heavily regulated across the globe. You'll find there are different regulatory bodies for nearly all the countries, and I think among them FDA, EMEA, they all kind of stand really at the top notch. They define standards which are kind of quite high, and they're quite well respected. But at the same time, as a site which manufactures a drug product, doesn't matter if it's chemical or biological, there's a lot of regulatory authorities there's a lot of regulations that need to uh, in a different way. If I can rephrase this, a manufacturing site needs to be up to the standard of the regulatory authorities, not only where it's located. So if the site is in Ireland, it not only has to satisfy the regulations of the authority in Ireland, but it also has to suffice the regulations where the drug is actually being marketed. So if it's marketed in US, then it would be FDA. If it is marketed in Japan, then it would be PMDA, or say likes of India, then CD's SCO. So it's a very heavily regulated industry, and biopharma is again a complex manufacturing process. And as a site, it requires different clean rooms or clean environments that again feeds in a lot of components, a lot of consumption of energy, a lot of equipments, automations, so that it can remain in a controlled state. And that's why all these factors increase the cost of the product, increase the cost of manufacturing. From our site point of view, if I want to explain the complexity and how much is invested, only this building and this building is are just the manufacturing or the core manufacturing areas. These are clean rooms. They're maintained with positive pressure and ensure that the air is circulated and it's filtered so that the particle, I would say the amount of particles that are in the air are up to a certain level. And these are kind of defined based on ISO standards. So you have ISO 7 level rooms, clean rooms, then ISO 8 level clean rooms, and based on how or like which processing is being done, different rooms have different ISO levels. Just from a drug, the cycle of a drug, how it kind of what's the timeline from how it's been discovered and how the journey happens. It's really a long journey. It's not as simple as the chemical drugs because there's a lot of work involved. If you look at when I spoke earlier about different strains available, you can find different strains. It can actually give you different antibodies which may be of interest, which may not be of interest. You can get like say 10,000 to 15,000 compounds that can help you fight your disease, but out of those, only few of them would be something that are of interest, that can potentially show that yes, these are the ones that can really fight or say kill the disease cell, or they can actually work effectively to that particular antigen. It takes almost 3 to 6 years for research and come up with those compounds and say like, yeah, A1, B6, or say Z5 strains are the best strains, and these are the ones that can be used potentially for clinical trials. And that's when the clinical trial happens. It's been administered to patients who are volunteered. It goes to a different dynamic or a larger group, phase 2, phase 3, and phase 5. And based on the results, based on what outcome is achieved, the drug then can actually go for a new application. The interesting thing about our site is that it not only has a commercial manufacturing suite, but it also has a clinical suite. And what clinical suite does is it actually manufactures drugs for clinical trials. So right now we probably have almost 10 to 15 commercial products which are sent across the market, which are sorry, which are approved. But we also have almost 30 to 35 new products coming in for clinical trials that we need to manufacture, and those drug substances then are sent for clinical trials. And if they are if they're approved, if they reach to phase 5, and if they're good for application, then they reach back to the site and say that yes, we have a facility that we can manufacture in large scale. So that process can take 6 to 7 years or even 10 to 15 years. And out of those 32 or 35 drug products, it's not certain that every drug product will reach this phase. It could really depend on the stability. If there is 1 or 2 cases that it has severely impacted the patient, then that drug can be pulled back and say like, okay, there's a patient risk, so we shouldn't go ahead. So there's a lot of risk involved. Once that new drug application is made, then there are trials, there are engineering runs. There's a scale up, and based on the facility that manufactures, we need to send a regulatory filing to the different regulatory authorities where we'll be catering the drug, and just wait for their approval. So in 2019-20, there was a mega expansion that was happening for new product that came in, and we finished the suite, we finished the facility, we did trials, we made pre-approval batches, and those batches were used to file for regulatory approval. And I think it's almost 6-7 months now that we are just waiting for the regulatory authority to come back and say that yes, everything is okay, everything is what it's supposed to be, and it's now allowed or being authorized to send to or to commercialize. So it's not easy, or it takes years and years of effort for the drug to come out, and just to appreciate that during the COVID or during the pandemic, I think this entire span was cut short into 1 or 2 years, or even less. And I think the amount of effort, or the combined effort that went through, was really, really huge, including our company, including a few of the colleagues who were responsible at not at this site but at a different site, and they worked hours and hours, days and night to get that through. So it's a very tiring process. And at the same time, there's one more thing I wanted to emphasize. Sorry, just skipped from my mind, but if I remember, I'll just come back. So in order for me to just explain you that what goes into, or how that manufacturing works, I will just explain you the key manufacturing phases, and I'll keep speaking about how the manufacturing process, or how the bioprocess works, or how it's been manufactured. Okay, so in the next slide, every biomanufacturing process is nothing different to a fermentation process. The reason behind is that when we get the cells which has the protein of interest, or which can generate the protein of interest, I should say that, is delivered to the site. The Eureka strain, I say the best strain, is stored in cryogenic storage, or at very low temperature, so that it remains in a dormant state. And those cells then needs to be grown or cultured, and that will actually produce the antibodies or the protein drug of interest. And then afterwards, we harvest that protein, we purify it, because it'll have a lot of impurities, it'll have a lot of different proteins as well. We'll purify it, and till we get only one, or only the protein of interest that we are that's going to be our the active ingredient in our drug product. And then and then the final thing is the fill finish, which our site unfortunately doesn't do that. Our site is just a drug substance manufacturing site, while a drug product manufacturing site would be something that would take the active ingredient and will convert into a drug product. Say, for example, it'll have vials for anyone to inject those antibodies, or if it's a chemical drug, then it can be actually converted into different forms, like syrup, nasal sprays, or creams, or tablets, capsules, wherever that drug has been used. Right. So I'll keep going, and I'll keep explaining you about each and every phase, and but I will not go much into more technicality, but I'll make an effort to help you understand the entire flow, and I hope I'll reach up to Rohit and Navneet's expectations. Okay, so upstream. Why it's called upstream? Because what we are doing here is we're growing cells. We are just trying to use basic fermentation technology to enable the cells to grow and reach to a certain cell density, so that it starts generating the antibodies. It starts generating the protein that we are really interested in. Okay, it's done in a controlled fashion, and obviously to grow cells you need nutrients that's fed in form of a media, and to keep them healthy and keep them viable, it's fermented in a controlled manner. It's similar to how you make bread. It's similar to how you brew beers, or any other drink, or any other fermentation process. So it starts from a cell vial. Then we do a gradual increase of gradual growth of cells. If we do a drastic move from a vial to a bioreactor, or say a larger volume, two things can happen. Well, first thing is it kind of it gets over nutrient, so the cell may kind of reach to their death phase quite fast. Or second thing can happen, that it may not actually be able to grow, because it's over fed with nutrients, and eventually it won't be able to give us the right amount of protein. So that's why there's a gradual process of increasing the cell number. Say from a vial, it'll go first to say 100 to 150 ml of flask, and then it'll kind of be transferred. It'll be incubated for say 2 to 3 days. They'll check the cell density, and then they will inoculate in a higher volume, say a 500 ml flask, and then afterwards it'll go back again to a 3-liter flask. After that, in order to increase the volume, there are a couple of other couple of things that can be made use. One is a wave rocker, or the other is a single-use bioreactor or a small bioreactor. Then afterwards, again, going in progression, bioreactors can be from 50 liter to 1,000 liter, and it can progressively reach up to 15,000 to 2,000 liter capacity. That is upstream. And few of the major differences in upstream is the way how the bioreactors work, or the way how the cells grow. One is the batch process that everyone is made, it's quite popular, but fed batch and perfusion are the two quite popular fermentation methods that are used in biopharmaceuticals. Fed batch is something that you keep feeding the media, and the cells kind of keep growing, and they're kind of at a certain phase, and then afterwards, in one go, you extract everything out of the bioreactor, and then you process ahead to get your protein of interest. In perfusion, what you do is you remove certain cells, you take out your protein, and then you put back the cells again in the same reactor, and you keep feeding more media so that they can actually grow again. They can actually generate the same amount of protein, and then that cycle repeats. So fed batch is that gives you a very large batch, high titer value, but perfusion gives you small batches, but in a more continuous fashion. So that's why upstream processes sometimes it does vary, and I think from product to product, depending on what kind of product we are manufacturing, they do vary. In our site currently, the old site or the existing site uses perfusion, while the new site uses a fed batch system. But there's also certain trials that they're using a combination of perfusion on a fed batch bioreactor. So that's something very interesting, that they're trying to stretch out the capabilities of an equipment. That, okay, how can we actually, we have a car, how can we actually make it like make it a bigger car, or how can we actually make it run faster? So all these possibilities are currently under trial right now. So from upstream side of things, we've grown our cells. We made sure that they've made enough protein for us, and the cells are kind of, we are saying that, okay, they did their job, they have the proteins ready for us, and so now it's the right time to harvest them. All right, so that's the next phase, or the critical phase of harvesting the cells. Depending on where the protein is being generated, in majority of cases, the proteins are actually present inside the cell. That means we need to break the boundary of the cell, or we need to break the cell and extract the protein. Or sometimes it's actually present around the cell, so it's present outside the cell wall or the cell entity. So depending on how and where they stand, centrifuge and depth filters are two most popular way of recovering or harvesting the proteins out of the cell. I won't go much in detail because everyone knows, everyone would be quite familiar with the concepts of centrifuge and also depth filters. Depth filters essentially are filters with different membranes, and which allow to pass the proteins, and while the cell debris stay within the membranes, and they can be taken out. There's another method called ATF, which is called alternate tangential flow filtration, which is ATF. I'll just move back two slides. So when I mentioned about perfusion, where the cells, so you recover the proteins and the cells are put back in, it primarily uses an ATF filter to do that. This diagram shows that when your cell density reaches to optimum level, you push your cells in, and these has filters which are as thin as a straw, and they are in a vertical fashion. Proteins tend to ooze out from the filter, so the permeate is sent, the blue line that you see is sent to a recovery bag. So that's your core harvest where the protein lies, while the cells are still stuck inside the membrane. So what it does is that it pushes using air back inside the bioreactor. And what happens then is the cells are still active. You feed in more media and say that, okay, come on, we're giving you more media. Now we need more protein. So that fermentation, they're keeping the cells at a very at a stage where they can provide protein, but they don't allow them to go to the death phase or the lag phase. The death phase, which we do not want. So that's why the perfusion fermentation, it keeps on going for say 30 days, or even in some cases I've seen and I've heard that it even stretches to 90 days, and you probably get in one batch. That means in one entire process of fermentation, you can easily get up to 15 or 20 batches of your harvest. All right. So we got the harvest, we got the protein, and now the next step is downstreaming or downstream. So downstream is nothing but purification of proteins, because there are a lot of cell debris, there are a lot of other compounds, different proteins as well, different cell components after rupturing them. So all these are not necessary. They're not required. We're only required, only in need of that specific antibody or specific protein that's going to be our active ingredient. So downstream process is essentially, I would say, the most complicated and the most crucial step within the biopharmaceutical manufacturing site, because that involves a lot of purification, and the purification steps, if something goes wrong, there is no recovery back. In certain cases you can, but then if they're not purified, or if they're not achieved, then either at the end of the batch or the final filling, your yield is either very low, or there can be potential contamination which would impact the patient directly. So what happens in downstream purification? We are purifying proteins, and two major concepts or methods used is chromatography and filtration, or the membrane filtration. These are the two main concepts, and they're essentially common across all the manufacturing or biopharma manufacturing sites. The main goal, apologies, there's a typo there. The main aim of purification is to remove impurities and inactivate viruses, and also ensure that while purifying the protein, the structural integrity of the protein is also maintained, because at the end we don't want protein to denature that can actually just not be of any worth to the patient when it's injected. From a fundamental point of view, chromatography is just a separation technique or a filtration technique based on, it can be based on size, it can be based on affinity. It can also be based on a certain um, charge. Sorry, that was the word. So it can be based on different charge as well. So size exclusion is the most common and the most simplest form that's being used right now. And I think that are used for simple proteins, but they may not be used for, they may not be more popular commercially. What it does is it has beads, so that resin or the column is packed with a certain type of compound, and when you put in your eluate, so when you put in different compounds and proteins, what it does is it kind of travels based on gravity, and any larger molecules will pass first, and any small molecules may be sorry. So they'll pass actually at the later stage. Ion exchange chromatography, it's based on the charge. So again, it's a biochemistry side of things. So I apologize if I'm not saying the right term, but based on the active binding site of the antibody, and depending on what is the functional group, they have a specific charge, and based on their affinity to a negative charge or a positive charge, you can actually retain, or you can pass your protein, or you can clear off your impurities based on different elutes. The last one is the affinity chromatography. This is interesting because it uses a specific ligand, or it uses a specific antigen that kind of binds your protein. It's more like trying to capture your protein of interest and let everything else flow down, let all the impurities run away. And then afterwards, after adding a different type of buffers, which are essentially solutions made up of different base and different different salts and different acids, and based on that, you can actually recover your proteins back. Then afterwards, viral inactivation and filtration, these are crucial because chromatography cannot exclude or cannot remove viruses. So what essentially is done, and what popularly is done, is you use a viral inactivation method. You try to denature the viral protein by using lower pH, or say higher base, but also ensuring that you're not impacting your protein of interest. So there are different hold times where you increase the pH of your yield, or your eluate that you get, and then afterwards, once you are sure that you've kept your hold, then you just you change your pH back again, and that will kind of help to maintain your protein of interest not to denature, right? The other method is viral filtration. So depending on how the sequence goes, your viral inactivation can actually be at the very start of your purification, or it can be in the middle, or it can be at the very end. But viral filtration is actually being set up at the very end of your process, because once the protein goes through different purification stages, when you are finally able to get a protein that is pure enough, then you need to make sure that it stays in the pure form, And if there are any potential viral proteins present, you can actually filter them using viral filtration. It uses nanofilters to ensure that any viral particles or viral proteins are being filtered out. Yeah, sorry, I forgot about ultrafiltration and diafiltration. So just to recap, you have your harvest. You're putting your harvest through different columns. That means you're trying to purify. You're trying to reduce the concentration of your impurities, but you're trying to increase the concentration of your protein. So while doing that, your amount of protein starts reducing, and you reach to a point where you feel that the protein concentration is at the highest and the purest form, but still there's a potential contamination. How can I get that out? So ultrafiltration is utilized to actually reduce those compounds. And this diagram shows that it's a tangent again utilizes tangential flow filtration so that filters don't get blocked. And here the permeate is actually your waste, while the retentate is actually your protein that stays up. But the problem is that once you get a higher concentration, it's not in the best medium that can be injected directly to the patients. So what you do is you use diafiltration. Diafiltration essentially is a form of, again, increasing the concentration of protein, but it may be also changing the buffers that are, or the medium that's currently what it is in, because you can't inject a highly acidic or highly basic protein compound or something directly into your bloodstream. So you need to make sure that your the pH level or the electrolytes are essentially similar to your bloodstream. So that's why you use ultrafiltration and diafiltration. And finally, that's when all the retentate is your actual protein of interest that you achieve. And the last step then is fill finish. Now, in certain industries, purification also involves a last step, which is called formulation, because as I said earlier, that our site is it's actually just a drug substance manufacturing site. It's not a drug product manufacturing. So everything is bulk filled and sent to a different site. So in order for the proteins to stay stable, there are different buffers being added to ensure that there's a certain shelf life, to ensure that if there's while transportation, all the protein is actually being frozen. So it kind of goes in, it's still kind of put into severe environment. So when it's thawed back and when it's ready to be filled into vials, it ensures that there's no structural deformity, so that it doesn't lose its ability to bind to the antigens. So if there's any structural deformation of the protein, then it's just useless to administer. So the final fill finish, then it can be into two different ways. If a site has an ability to have a fill finish section, then that product can directly go into vials after formulation. Or if it is something like our site, then it goes into bulk filling, and then afterwards it's shipped across to different site, and then it's again reformulated and filled into vials. And these vials then can finally be administered to directly into patients. Again, that dose or the dosage volume is all dependent upon medical experts and hospitals and doctors, I guess, to whom we are catering this drug to. I think I hope I am able to explain you what goes into manufacturing, and I'll just do a quick recap. We had cells which had a protein of interest. We used fermentation to increase the cells to a level that we can extract them. So we harvested them. We took out the protein by removing the cells or by breaking the cell, but we had a lot of impurities. And what we did was we went to different stages of purification, and we found the purest form of our drug. And that purest form was then used, was formulated so that it can go into final vials and reach to the patient. This process is common across any biopharmaceutical site, including vaccine sites as well. They might have slightly different unit operations or different stages or different additional steps involved, but whole and soul, the core of any biopharmaceutical drug manufacturing are these 4 steps or 4 phases, and I suppose that is the whole part about manufacturing, drug manufacturing. The last thing what I want to emphasize is the use of disposable technology in biopharmaceutical, because although it's regulated, although it has evolved a lot, there's a high trend of using disposable technology in biopharmaceutical, and some of the main reason and why is because it gives more flexibility on different product changeovers. You don't need to sterilize it, or you don't need to go through a cleaning process, and you don't need to involve quality to assess that it's correct or it's not. It avoids the risk of any potential contamination as well. And because their assemblies, they can be retrofitted into equipments, that means that if there's a high turnover or if there's a high changeover required for products, it can be done quite easily. To be honest, disposable technology can literally replace all your equipments, all the standing equipments in your site, because some of the companies, especially at a very small scale, can utilize disposable technology right from the start of recovering the cells, growing the cells, till the final filling stage. And I'll show you in the next slide how it does. But a bit of downside about using disposable technology is the fact that you don't have control over those assemblies or parts. You're always dependent on the vendor. And if there's a shortage of supply, your production is at standstill. And if it's at the stage where it has crossed your harvest, you have no choice of holding that, and you just have to drain the batch. And when you drain a batch of a capacity of 15,000 liter bioreactor, it could easily cost from 5 to 15 million US dollars, if I want to just give an approximation. It is very, very, very expensive to manufacture. So that's why there is a huge emphasis about quality regulations, and even how about different controls. If I go one slide down, that's what I was speaking about, that that a single-use technology or disposable technology can start right from your upstream stage, where you have different bags being used in to slowly increase your cell capacity or cell density. It can actually be sent to bioreactors which have bags that have disposable agitators. It could potentially go more than 500 to 1000, even 2000 liter capacity can be harvested. Even there have been talks about using disposable centrifuge as well, which I'm not familiar, but I just heard that that is possible. And then again, you have filters made up of single-use filter cartridges, so that you don't need to use them again, or you don't need to go and clean them and use them again. You use once and you can dispose it, and you can use a fresh one. This just gives an overview of how disposable can be useful, and how they are kind of now becoming an essential part of everyday manufacturing. Now, I might have missed that disposables are actually assemblies. They're kind of irradiated, so they come as sterile products on site. And all we do is we just ensure that they are actually being attached or assembled in a sterile environment, and essentially that's how they're being used in the manufacturing. Last thing is last two slides. I promise I won't delay much. Are the challenges. Pharmaceutical environment is heavily regulated, so anything that you do, any changes that occurs on the site or on the manufacturing floor, say the process, say the parameters, say the way you test, or to assure that what test you're using, everything has to be recorded, and everything has to be done through a change control, as we say. And if it requires and if it affects the quality of the product, then the product kind of steps back in the approval stage. So there has to be changes made in the regulatory approval, and a lot of inspections happen, and a lot of documentations are involved. So there is a huge work involved if even a minor change is required. However, every company or even the regulations do give you that flexibility of where you can draw a line saying that that X amount of changes are acceptable, and where you can where it says that no, these changes are not acceptable, and it'll kind of impact. So all these are kind of daily challenges. The second and the foremost, which has hit all the industry, is COVID and the global pandemic, because not only it reduces the amount of individuals working on site, but it also impacts the suppliers. So if you're actually going straight to disposable technology, we are just trying to we're just relying heavily on those suppliers, and if the production capacity is reduced at their end, then it's going to impact us or impact the site for supply. There's also an interesting fact that because globally a decision was kind of made to support vaccination, majority of the disposable assemblies were actually diverted to the vaccine manufacturing sites. And at the same time, I don't know if it's true, but there was an executive decision done by US saying that any, say, pharma company or vaccine company who is registered in US, they would kind of get a preference. So any drug that's been manufactured has to go there. So all the political involvement and all the focus on the vaccine also kind of diverted the the resources and supply towards vaccine sites rather than actually coming to rather than the regular supply. So that was also a huge hit considering the pandemic. And likewise, material shortages that material shortages, not from a perspective of the manufacturer, but a perspective of depleting resources, is again a huge concern going forward as well. Also, as a site, we have a lot of clinical trial products coming over. So there's a huge changeover coming in on site, and as a result, a lot of resources are being pulled in to troubleshoot at that point, and also manage the routine manufacturing. So if you consider my own situation right now, my challenge was that I was burned out from last week, working almost 50, 60 hours trying to work from home, be on site, and share the challenges that are happening on site. And my biggest challenge was that how could I make this presentation better if I had more time. So this comes to the end of presentation, and I hope you get something out of it and appreciate that there's a lot of similarity between the biopharmaceutical drug manufacturing and normal fermentation, which you might have studied in your academic time duration, or even in your current work profession. So that's all from my side. And if there are any questions, and I said to Rohit and Navneet that I'm just a presenter, please don't shoot me.
Thank you, sir. It was really a thorough presentation, something which is like it's like almost attending a classroom textbook kind of presentation from a person who is really deeply involved in pharmaceutical industry. It was a pleasure to listen to you. I think there are a few questions.
Yep. I
Sneha?
see this.
Sneha, could you ask your question?
Yes, ma'am. Hi, sir. How are you?
I'm good, Sneha. I read your question there. I can
Yes.
answer that straight away.
Thank you, sir.
Sorry, Jashi, can I request you to just mute, please? There's an echo heard.
Is it okay, sir? Can you listen to? Hello?
Yeah, that's ideal. Yeah, that's good.
Okay.
So during downstream manufacturing, after every after passing the drug product through every column, or say different stage of column, sample is being taken to test for bioburden and endotoxin, and those results assure us that if the batch is contaminated or not. The second check is that after every step of column, or say after every column, there are filters to filter out any potential contaminant as well. So that kind of gives you a check on your contamination as well. Does that answer your question, Sneha?
Yes, sir. Thank you so much, sir.
Okay, thanks.
Yes, Ashutosh, it's
Yes, sir.
very nice and elaborate presentation.
Thank you, sir.
I am curious, can you just a little bit elaborate about the cleanliness? How do you ensure after a batch is produced and before next batch you go? Because
Yes,
here
sir.
in pharmaceutical industry, we have to ensure a very thorough cleanliness, nothing should be remaining. So what are the various techniques which you use or pharma companies normally use to ensure the cleanliness of the equipment as well
Yes,
as
sir.
the facility?
Yeah. So I'll answer that in 3 steps. So the first is the facility. facility itself, because it's a clean environment, restriction of there's a different company that takes care of sanitization of the walls, of the floor of the area. So that's taken care, and there are certain regulations behind that. So that's been done on daily basis, and a deep clean is kind of done on maybe quarterly basis or every 6-month basis. um, Equipments, inside the equipments, if they are standstill equipments like bioreactors, there is the cleaning is done through CIP, which is clean in place. So there are it goes through 2-3 different cycles. First cycle is, say we use high pH acid,
No, that's
and
that's
then we use,
Ashutosh, that's fine. In fact, that is.
yes.
But my question was, I wanted to know how do you ensure the effectiveness of
Ah,
the cleaning? That is whether the
okay. Sorry.
CIP you have followed.
Yeah,
Okay, whether CIP worked properly or not.
yeah.
That
So yeah, I get it. So sorry, apologies. I
no
got it
problem, no
in the other
problem.
way. So it's ensured by first by automation controls. We ensure that the cycle that's been already validated, if the cycle has ran into completion without any stoppage or without any interruption, that's the first assurance that you have. The second assurance you have is by visual inspection, because the reason I'm saying visual inspection, because prior to confirming that this is the right CIP cycle, as a validation engineer, we go through different validation runs. We take different samples inside and from the CIP when it's running. We ensure that those cycle are capable enough to clean it. The test samples that we get, the results meet the specifications. When they are validated, then we say to the production that yes, this cycle is appropriate. This cycle is absolutely perfect to run. So automation controls and the cycle runs, they ensure that it's clean correctly or not. Alternatively, samples can be taken as well, which can be sent to the lab, and they will assure us that from a chemical residue point of view, if the TOC level, do they meet the specifications or not. So that's those are the two things. And again, just to add on, sir, to your question, after cleaning, the vessel also goes through steam process, which is called steam in place. So steam in place, again, which I am currently involved now as well, also assures that the lines and the vessel is not sterilized, but it gives you a sterility assurance level and says that after this SIP cycle, there is least chance that a contamination or a living organism can grow or stay there.
That's fine. My second question, of course, out of curiosity, because this is a very, very important topic you are talking about.
Yes,
Like
sir.
you are dealing with food, the pharma is also equally very important. So, like in the food, there are clinical trials. We go for evaluating the efficacy or effectiveness of a biomolecule or food material. Okay, so in the drug, how do you do the effectiveness of the drug? Whether the drug for the purpose of which it is manufactured, it is really working or not? How do you
Yeah. Yeah.
test?
No, sir, very good question, because I think like every drug product, sir,
There must be, I am sure, there must be some instrumental method, chemical method, and
yeah,
all those things. But the
that
efficacy,
is
risk,
that,
other risk factor is involved, and all those things. Is it done
sir,
on
that is
human subjects or?
Yes, sir. So in order to check the efficacy, sir, that's why, sir, clinical trials are done. So I will just go back to that slide, sir, which I showed. From Janssen point of view, sir, the clinical trials are actually done on different subjects, or I should say, human trials are done. So there are
How
volunteers.
you select the subjects for the clinical trial? That is they have the
So
hospital.
they are. Yeah, sir. So I think from I'm not 100% sure how that works, but from my knowledge through different speakers, what they mentioned is that subjects or volunteers, I would say, in a very respectful manner, they would be coming from different institutions. It could even happen that a certain time that a patient who is severely ill and does not have uh, any hope may come forward and say that yes, I can actually go into a drug trial program, and that's why it's been done first to a very small group. That's why in the slide it says 20 to 100, and then afterward, once that efficacy is shown based on the responses, it goes to a different higher group. And when we say higher group, sir, higher group means more subjects, but at the same time, it can also be different demograph as well. Because, say, as a protein drug, my immune responses may not be the same as someone living in the US, or a US person who has lived in US. So I think that's why, sir, 6 to 7 years is the time frame where these trials happen. When the efficacy is really high, then only it goes forward to be applied as a new drug. So it's a very lengthy process, sir,
Yes, yes, yes.
and I think I just to add on to your query, and from my understanding that this phase was fast tracked for the vaccine, for COVID vaccine. So I think, uh, and I suppose, in order for companies to fast track, they base the vaccine from a previously, how do I say, from a previous vaccine that was already effective on a similar kind of virus. I think it was based on influenza virus.
I think that's fine. Yes, very good. Any other question from other people? There must be, I am sure.
Thank. I don't have a question, but I just wanted to know your experience since you have worked on a, on a food side and now on a pharmaceutical, and that too in a very big organizations and at the manufacturing level. So how does you rate both the experiences, and what are your experiences and the challenges that you face, and which one you prefer to, I mean, not to prefer, but overall show your experience to the audiences, especially the young audiences around here. If you want to, kind of give your thoughts on that, it will be really helpful.
Yeah, sure. See, I'm just 4 years in this area, so it's a very vast field. Um, to sum it up, I think there are a lot of similarities between both the fields. Food is also regulated. Pharma is more severely regulated. So for those who are kind of more interested to work, or who want to work in a very regulated environment, and with constant changes happening around, I think pharmaceutical gives you a big opportunity. And from my personal experience, I feel that um, I feel food industry is again, that has kind of expanded a lot. So am I saying this correct, Rohit, or can you just maybe repeat the question, and I'll just try to answer in a better way?
No, no, I just wanted to have your views. Like, you have seen both the sides, food and pharma.
Yeah.
How do you rate the experiences that you have?
Okay.
And if you have to give any suggestion to the young audiences here, what would be that?
Okay. To rate the experience, or I would say, I enjoy working in the pharma industry because it's highly regulated, and it shows, again, it company to company depends. It shows that we are kind of working towards an end goal, which is to help the patients. So, because what you're doing is you're actually making a drug that can actually give hope to people. Doesn't matter if it's like a chemical drug or a biological drug. It doesn't matter. But all you're doing is you're actually giving a slight bit of hope to people that they can still go ahead and continue their life on. So I suppose it's from my experience, I feel that this job has kind of given me that peace that I'm doing something good, something better. But at the same time, it doesn't mean that food industry or is something different. Actually, I think Mishra sir would know the name of the person, Dr. R.K. Jain. If you would know him, he was an alumni of IIT Kharagpur. He happened to be a principal in AD Patel Institute of Technology,
India,
and
yes,
initially,
he was a principal scientist here and research officer in our project
yes,
society. We are good friends. Yes.
exactly. So I still remember his word when he came the first day on in our engineering class. He said that you guys are future engineers, and you're doing a job. You're doing God's job. You know, it's a bit cheesy to say, but you're doing a very godly job, because the nature has given grains or wheat, and you're actually converting that and giving an edible form of food to people. So I feel that both food and pharma industry, they both serve a purpose at the end. It all depends on your personal choice, how you want to see it and how you want to build yourself. A good thing in food industry is that it's sorry one of the good thing I liked about pharmaceutical industry is that it kind of sees the diverse skill set, because it's not just core um, biochemistry, it's not just core chemistry or chemical engineering. It's a mixed bag of skills that you require. And while in food, it becomes very specific knowledge that the companies are looking for. So sometimes you need to make a choice where you want to go. Do you want to be in a field that you want to be very specific and expert, and or you want to choose an industry or a field where your expertise can be built over a period of time, and still you're able to kind of see yourself growing professionally and utilizing your transferable skills. So I hope that that answers and gives some bit of encouragement to anyone looking forward.
Yes, it does, Ashutosh. Thanks for sharing your experience. Just to roast you, a question to you, what do you go with? Let food be your medicine, or let medicine be your nutrition? Tricky one for
Let
you.
food. Yeah, I can actually. Let food be your. Can you say that again?
So let food be your medicine, as we always say, Hippocrates says, or
Yeah.
what I recently just framed up, let medicine be your nutrition.
I think
What do you go with?
Rohit is trying to say Rohit is a food company. So if he does his job good, Ashutosh will not be required. है ना? Ashutosh is
I,
required because Rohit is not doing good job.
I
If you provide a good food, people will be always healthy. They will not fall sick.
Sir, you
it
have
is
added
it is
spices.
Huh?
I was
I think,
roasting him and you have added spices into it.
sir, it comes down to the question that which came first? Was it chicken or was it the egg? But I can answer your question, Rohit. I think I'll go with the Hippocratic mentality that food is your. Sorry, I'm just forgetting. So I'll go with the first statement,
Good.
and the
That
reason
means you're
why
still a food technologist.
I am indeed, and I still be a food technologist, but
We are
more than
happy to
that,
hear that.
I'll be an engineer.
Rohit,
Yes.
just to, since it is being discussed, I think a few years ago, all right,
Mhm.
4 5 years, there was some Nalini Singh, you know, that Nalini Singh, famous reporter, and
Mhm.
she used to report in that DD News, science fiction and science stories and all those things. So once I was listening and she was reporting there is a family in Malda in West Bengal, and she
Okay.
was telecasting the interview from that family, all right, and that family was claiming that for the 7 generation, no doctor has visited to their family, or no person from that family has visited to their any hospital or any doctor.
Wow, that's impressive.
And the person who was there, he was aged about more than 70 years or so, but he was looking so young as he was very about 40 45 years. And the reason he was, the family person was telling, they never used cooked food, and
Oh,
they
okay.
always ate only raw vegetables, fruits and vegetables.
Yeah.
टमाटर है, even gobi hai, patta gobi hai, brinjal hai, all those things. And they were telling, even if they have some relative in their family, so they will bring food from some neighbors or for somewhere to feed the relative. But उनके घर में कभी खाना नहीं बनता था.
Wow. They're taking the nutrition in the best form.
In the West farm, that
Yeah,
is
in the
raw.
best form.
I just want to comment one thing that I know I presented about biopharmaceuticals, and it's all looking bright future, and how it's trying to impact and affect people in a positive way. But I think food is the primary source, and the for our human body. It kind of depends on that. But I think when complications and when diseases happen, I think that's when
I think we have
using
some
using an, using
Sindhuja,
an
Sindhuja Maka. She has given some comment. Maka?
yeah, I'm also trying to explain that comment as well. That
Sindhuja is here. Can we listen from her? I think she is. Hello, Sindhuja. Sindhuja. Yes. How is
Hello.
Professor Maka and तुम्हारे मम्मी पापा कैसे हैं? Fine. What about भैया? She is a daughter of our colleague. अभी तुम लोग कहाँ पर हो? Where are you now? You are, I think, muted. Please, we cannot listen. We cannot listen to your voice. No, not now. Still now, we are not listening. We are not able. Uh, yes, Ashutosh respond. I think there is some problem with her connectivity. Mic.
yeah, no problem, sir. What I was trying to explain is that food is a fundamental necessity of the human body. It definitely needs for its well functioning. But I think when at the time when diseases like cancer, and which cannot be cured by normal nutrients or your own immune system, I think biopharmaceutical, it's more like a out of the box solution. I wouldn't say solution, but an alternative, that if we are able to have certain antibodies that can actually fight that, and it's not the fact that we need to keep taking them or keep injecting them. It's I think in future lies where we can actually use those antibodies to train our own immune system to fight back again those kind of diseases. Because the problem with cancer is, and I think many of us would appreciate or maybe agree with me, is that even though you remove a tumor, a cancer tumor, there is a chance that cancer can regenerate or redevelop. So some certain therapeutic or biotherapeutic compounds can actually help your immune system not only to fight against the existing cancer, but also trains your immune system that in case if a similar cancer cell develops, then your body immune system is able to attack it and destroy it. So that's the beauty of biotechnology, I would say, and hats off to his. And and I think we cannot compensate food with biopharmaceutical. So I don't know how to answer that, but no, we cannot convert a biopharma into a food company. It will
What
never
I
happen.
Rituja is taking, in fact, it has already happened. You see the Glaxo. Glaxo is a well-known pharmaceutical company, but still they are a well-known food company as well. They are manufacturing a lot of food products. So that is true. Many pharmaceutical companies are making food products.
Yeah, no, agree, sir. Pharmaceutical, nutraceutical, absolutely,
Yes.
and no doubt in that. And just to update that GSK was taken over by the pharmaceutical side of GSK was taken over by a company called Thermo Fisher.
Yes, yes. That
So
Yeah.
Thermo Fisher is again a supplier, but also now a manufacturer.
good. Anyway, but food and pharma, they are complementing
Bye
to each
bye.
other.
They
They
are
say
bye bye.
good, very good. I think it was very elaborate session, almost 2 hours.
Yeah.
Thank you, sir. Sorry for that much time. Uh, but thank you for listening, and I really hope. And if there is any more further questions, or if you want to reach out to me, please feel free, sir. I personally want to say thank you, and again, I was quite introvert and in my bubble, but for good reasons. It was just the time when I faced a rock bottom. I had to just focus on myself, but it doesn't mean I am disconnected. I'm always connected. And it's almost 3 years, sir, that I haven't seen my family in person. So I'm really hoping if things change, I'm able to go back home and meet my family, and even make time to meet yourself and
And
my
that you
dear
must do
friends.
now. That you must do now. In fact, people are coming from about even. In fact, my daughter is there in Frankfurt,
Yeah.
and she regularly. Last year also she came during December, January. Now also it is coming. She is coming this month. So थोड़ा सा ये रखो ठीक है बाहर रह रहे हो अच्छी बात है बट डोंट फॉरगेट योर पेरेंट्स
Never, sir.
फॉरगेट
Never, sir.
योर
Never. Absolutely never, sir.
एक बार तो साल में विजिट कर लिया करो दैट इज माय
No, sir. 100%. And sir, it is for their own safety. क्योंकि sir, it's better to be more
ये यार कुछ नहीं
safe than never, sir.
ये तो अभी आई थिंक वी हैव टू नाउ अगेन ये कहेंगे कि फार्मास्यूटिकल कंपनीज कुछ तो और कुछ मीडिया वाले नॉट ऑन द रिकॉर्ड आई थिंक रिकॉर्डिंग दे हैव स्टॉप्ड सो कुछ मीडिया वाले कुछ फार्मास्यूटिकल कंपनीज वाले हाइप ज्यादा क्रिएट किए हैं तो otherwise now we have to be take it as a go normal don't panic what our prime minister says
Yes, sir. It's going to be normal. It's just a matter of time, and just yourself.
we have to feel confident yes कि ये सब ठीक है जितना डरोगे उतना ही इसमें और
Yeah.
Sir,
Come soon,
100%.
thanks. You will be fine. Yeah.
Huh?
I'm saying come soon here. You'll be fine.
come to India. एकदम से अपने आप
Yes, sir. I need Indian spices, not.
कम से कम अपने पापा मम्मी को बहू तो दिखा दो एक बार.
अरे, no, sir. They did visit me in 2019.
Yeah, they visited. Okay,
They visited, but then afterwards I did not reach home since then.
very good.
So last time I was home was 2018, sir. So after that, I haven't got a chance to come home.
Good. So Jayashree,
Yes, now I think we are coming to an end of the session. So I would request our master student Pallavi to give the vote of thanks to Ashutosh sir. Pallavi.
Um, Hi, ma'am. Actually, Pallavi is not available, so
Okay, okay.
uh,
Sakshi. So
that's Neha.
would you
Sneha,
do
it
the
is Sneha.
honors? Oh, Sneha, sir, I could not see. Okay, okay. Sneha. Oh, sorry.
Yeah. So, okay. So, a very good evening to all. It gives me immense pleasure to deliver a vote of thanks for the FCTL webinar series. And my special thanks to the speaker, Mr. Ashutosh Tank sir, for giving a unique and interesting introduction about his place of stay as well as his work, and a brief overview on the biopharmaceutical drug manufacturing and how biopharmaceuticals have huge potential with lesser side effects than the chemical drugs, and the challenges it's facing currently, and giving in-depth knowledge on the key manufacturing phases like upstream, harvest, downstream, and fill finish, and the disposable technology. I take immense pleasure in extending my sincere thanks to our coordinator, Professor H. N. Mishra sir, because of whose efforts we are all able to get immense knowledge from different areas of studies. And I extend my thanks to one and all who have helped to make this webinar series a great success amidst the pandemic. At last, I would like to thank every participant for their sincere listening and learning. So, thank you all. Have a nice evening. Thank you.
Thanks very much. Thank you. Thank you, sir.
Thank you, sir.
Thank you,
Thank you.
everyone.
Bye bye. Thank you all.
Thank you, everyone. So, to our next webinar is
Thank
on
you.
22nd. We will notify about the speaker soon. And thank you so much. Have a safe year ahead. Thank you.
Okay. Bye bye. Bye
Bye.
bye.
Thank
Thanks.
you.
Bye.
Bye everyone. Thanks.